To begin our discussion of genetics, we're going to be focusing on basic principles of inheritance as laid out over 100 years ago by a German monk named Gregor Johann Mendel. And because of the importance of his contributions to the field of genetics, these basic principles or inheritance patterns are often referred to as Mendelian inheritance patterns. So let's talk a little bit about Gregor Mendel and why he's considered to be the father of experimental genetics. He was a German monk, he was a scientist, he was a high school physics teacher, and critically important, he was really interested in inheritance and how the passing of genetic traits from parent to offspring occurred. And to understand the prevailing views that were sort of popular at the time that he did these experiments, we have to understand that most people who were interested in inheritance viewed it as a form of blending inheritance, is what it was called. And in that model, the contributions of each parent in sexual reproduction would be sort of irreversibly blended in the offspring. You could imagine this like blending the colors of two paint cans, and once you mix those two colors, you really never get back the original colors in that mixed can. So that was the way that most people viewed inheritance, and that was the model that Gregor Mendel set out to test and to investigate. And to do so, he did a lot of experiments, over a decade of very carefully controlled experiments using pea plants as his model organisms. So these are just sweet pea plants like you'd find in any country garden. But importantly, they have characteristics that make them an excellent model system to use for genetics. So, number one, there were a lot of different strains of pea plants that had different, very easily observable, genetically determined traits. So things like flower color and seed pod color and plant size and plant length and flower position and all sorts of other very easily observable traits. And number two, it's very easy to control the mating or breeding of individual pea plants with one another. Although pea plants can self-fertilize, if you remove the male gamete producing organs from the flower before they mature, then it's very easy to control who's pollinating who. You just go out with a paintbrush in the garden and pick pollen from the plant that you want to serve as the male parent contributing the pollen, and brush that onto the female plant, which has the eggs, and so in that way you can very easily control who's breeding with. A third reason that pea plants are a really good model organism for genetics is that they have a very short generation time. So you don't have to wait forever to get your offspring from any cross that you've set up. You get them in a matter of, 60 days. And the fourth reason is that pea plants produce lots and lots of offspring. So with one cross, you're going to get hundreds of seeds that you can then analyze as the offspring of your genetic cross. So in one generation, you can actually get sufficient progeny to get statistically significant results that you can then analyze and have a high level of confidence in the results that you're seeing. So Mendel did over a decade of very carefully controlled experiments studying the inheritance of these different traits in pea plants, and in 1866 he published this very important seminal work regarding genetic inheritance called Experiments with Plant Hybrids, which was virtually completely ignored at the time. Nobody really understood its significance, and it really didn't shake up the world of genetics and the natural science community at the time. He died in 1884 without his work ever really being recognized as for the importance that it really exhibited, and it wasn't rediscovered until the 1900s, early 1900s, by three different scientists who essentially replicated his experiments in other organisms and came up with the same results. And having done the due diligence and gone and done their literature search to see if anybody else had published previously, they rediscovered this paper and publicized it to the world, and really they're responsible for him getting the accolades that he now gets, and the recognition for how important his discoveries were. And so that sort of gives you a little bit of background as to why this is called Mendelian genetics and why he's referred to as the father of experimental genetics. His contributions are really, really seminal to the field. Okay, so let's talk about what he did. So the first question that Mendel was interested in addressing was this question of whether inheritance was exhibiting this blending pattern or not. In other words, if you took two organisms that had contrasting characteristics and you bred them together, in the offspring, did you get a blended characteristic that seemed to be sort of an intermediate of the two parents, and did that blending remain down through the generations? So to test this question of blending, he started with what are called true breeding strains of peas. And this is a very important term, and we're going to come back and define this more closely. But what this meant from Mendel's perspective was that this was a strain of peas that bred true for the characteristic that was under investigation. So for example, if you had a true breeding strain of peas that had purple flowers, if you allowed that strain to self-fertilize across many, many, many generations, you would always get offspring that likewise had purple flowers. So it's true breeding for the trait of purple. Likewise, there were other strains that were true breeding for the trait of having white flowers rather than purple flowers. So that's what we mean by the term true breeding. So initially he took these two strains of peas with this contrasting trait of flower color, and he crossed them with one another. He controlled the pollination and crossed them with one another and examined the physical trait or characteristic that he observed in their offspring. And what did he observe in that first filial generation or F1 generation? He observed that all of the plants had purple flowers. In fact, these plants looked identical to the parent plant that also had purple flowers. So it didn't seem like there was any sort of blending between the two parent strains at all. In fact, what he observed was offspring in the F1 generation that looked identical to one parent but not the other. So that didn't seem to support blending inheritance at all. But the critical evidence that was the nail in the coffin for this idea of blending inheritance was that he took these F1 plants and he allowed them to self-fertilize or to intercross amongst themselves. So all of these F1 plants are identical because they're all formed as the hybrid offspring between these true breeding parent strains. And then he examined what he got in that so-called F2 generation, the second filial generation. And here's what he saw. He saw that the white trait, the white flower color trait, reappeared in that second generation. There was no irreversible blending. He didn't get a sort of intermediate purple color that remained purple across the generations. In fact, he got back that white flowered trait that he began with. So no irreversible blending. And furthermore, when he counted up his results, he observed that he got this very characteristic ratio of traits in this F2 generation where 3 out of 4 of the offspring would demonstrate the characteristic of the parent plant whose traits were exhibited in the F1 generation, what we refer to now as the dominant trait, and 1 out of 4 would exhibit that alternate characteristic, what we would now call the recessive trait. And that characteristic ratio of 3/4 to 1/4 or 3-to-1 ratio, he observed not just with respect to flower color, but for 6 other traits that he was studying. So as I said, lots of different observable traits in pea plants: flower color, flower position on the stem, seed color, yellow versus green, seed shape, round versus wrinkled, pod shape, inflated versus constricted, pod color, green versus yellow, and stem length, tall versus dwarf. All of these are genetically determined traits in peas, and he did very carefully controlled experiments with each one of these traits using different true breeding strains. And just as he observed with the purple versus white flower color, when he did that cross, he got that characteristic 3-ish to 1 ratio. When he examined all of these other genetically determined traits, he got very close to the same ratio. And so the fact that Mendel had this preparation in terms of his education in math, he wasn't just a descriptive scientist. He didn't just describe in very general terms the inheritance patterns that he observed. He actually counted them. And because he had a background in mathematics, the numbers and the patterns that he observed in these crosses allowed him to infer what must be going on with genetic inheritance. And really, I mean, take a look at the numbers for how many different plants he scored with regard to these characteristics that he was studying. 705 plants that were purple flowered, 224 that were white flowered. Come down here to the seeds experiments, and we can see he's counting, you know, you must imagine by candlelight, yellow seeds versus green seeds, 6,000 yellow seeds versus 2,000 green seeds. So this was a lot of work that he did over a decade, and was able to work out the basic rules of heredity because of his mathematical analysis of the results that he obtained. So this classic 3-to-1 ratio that we observe in the F2 generation, starting with 2 true breeding parental strains. What was Mendel's explanation for that? Well, we now refer to his explanation as Mendel's first law, the law of segregation. Of course, he didn't refer to it that way in his publication. That would have been extremely arrogant, but that's what we call it, because the basic rules that he worked out and what we now call his laws are absolutely true and still hold true today. So even without knowing anything about DNA, without knowing anything about meiosis, Mendel was able to describe what was going on during the production of gametes, leading to genetic transmission of traits, really working with a black box and working just with the math of the system. So what is Mendel's first law? So Mendel's first law states that the units of inheritance, whatever they are particulate. They're physical things that are passed down through the generations, right? They don't go anywhere. They don't blend. They're individual particulate structures that can be passed on from parent to child, parent to progeny. Furthermore, each parent has 2 copies of whatever that unit of inheritance is. And during gamete formation, the 2 copies that each parent has are going to segregate from one another. So applying these rules to the outcomes that he observed in his cross of the purple flowered and white flowered plants, how do they apply? So here was his explanation for the results that he obtained. So remember that the parent plants that we started with, the P generation, were true breeding. Well, Mendel hypothesized that the reason that they were true breeding is because the 2 versions of their, what he called, unit of inheritance were the same. So purple flowered plants were true breeding because they had 2 copies of this characteristic for purple flowers, and the 2 copies that they had were identical to one another, and so that was all they could pass on to their progeny, right? The characteristic of being purple flowered. So we now know that this is actually referring to a specific gene in the genome and the alleles that are inherited from each parent. And so we might call these 2 versions of units of inheritance alleles, right? So we could symbolize those using a simple letter nomenclature, capital letters to refer to the purple flowered trait, and then our true breeding white flowered parent would be the same, right? 2 alleles that are the same, and in this case the alleles are the ones that code for or specify the white flowered trait. So in gamete formation, we know that we go from the diploid state to the haploid state, and each gamete contains only 1 copy of each gene in the genome. So with respect to flower color, all of the gametes coming from the white flowered plant are going to have only 1 allele in each gamete, and that allele has to be the allele that codes for white flowers, because that's all this white flowered parent plant has to pass on. Likewise, the purple flowered parental generation is going to be passing on only the purple allele in its gametes, and every single gamete is going to have 1 copy of this P gene, and the allele that's going to be present is the one specifying purple flower color. When we allow these gametes from these 2 true breeding parental strains to interact and fertilize, so this is fertilization we're seeing here, what do we get? Well, we get back to the diploid state, right? We get back to a diploid organism, and these diploid offspring have one purple allele from this parent and one white allele from this parent, and the trait that they exhibited was purple flowers. So at this point, we can define this characteristic that Mendel referred to as dominance. So we have a hybrid plant that has a hybrid genetic makeup, but the plant looks, in terms of flower color, exactly like one parent and not the other. And so in this case, we would say that purple, that trait, is dominant over white. So purple is the dominant trait and white is the recessive trait. White is recessive because in this first generation it recedes into the background and becomes invisible. But as Mendel discovered, we know that it's not gone completely, because if we allow these F1 plants to breed, we get that trait back in the F2 generation. So it's not gone, it's just receded into the background. And so we would say that white is recessive to purple. And that's just based on the physical characteristics that we can observe in this hybrid generation that has one allele of each type. Now, what happens when this F1 plant produces gametes, and those gametes are allowed to interact and fertilize to produce the next generation? Well, we have to remember that, again, we're starting with a diploid plant, but meiosis separates these two alleles from one another, and so we're going to end up with gametes that have either the purple allele or the P allele, but not both, right? Haploid gametes. And so in this case, we have two possible gamete types that can be produced. Half of them are going to have the purple allele, half of them are going to have the white allele. And if we allow plants at this F1 generation, all producing half and half purple and white alleled gametes, to fertilize and to cross-pollinate in every possible combination, we can symbolize and depict the potential offspring that could result from that cross using this grid structure that's referred to as a Punnett square. And, oops. Mendel didn't come up with this. This was actually somebody who came along later that came up with this, the Punnett square, as a method of depicting offspring ratios. But he described these results. What is going on? Go away. Go away. Okay, so what we're looking at here, so as we said, Mendel's law of segregation states that the two allele types in the parent plant are going to segregate during gamete formation and then come back together during fertilization to produce the next generation. And so that's what we're depicting here in our Punnett square. Along the sides of the Punnett square, we're depicting gametes coming from each parent, right? Remember, gametes are the reproductive cells, so this would be what we might refer to as the male plant producing the pollen or sperm, and this would be the female plant and her gametes, the eggs or ova that would be produced. So we put the two types of gametes that would be produced by each parent along the sides and then combined them in all possible combinations to yield our Punnett square, right? And this is depicting the types of offspring that are going to be possible by all these random combinations of the male gamete with the female gamete. And what do we end up with? We see that 1 out of 4 of the gametes we would expect to have two alleles that are the purple allele. 2 out of 4 we would expect to have one of each, and 1 out of 4 we would expect to have two alleles that are both the white allele. Now, that would be their genetic makeup, but what is their physical trait going to be? Well, we know from the previous generation that all of these hybrids that have one of each type are going to end up with purple flowers, and these match the starting parental generation that we began with that had white flowers, and so we're going to end up with that classic 3-to-1 ratio that Mendel observed. And so this is the explanation for the observations that he made with all of those traits, and that's described very nicely in his law of segregation. So the units of inheritance are particulate. Each parent has two copies, and during gamete formation, those two copies segregate from one another. Mendel's first law, the law of segregation. All right, so we can now distinguish and add in a little bit more terminology and vocabulary to describe these observations and outcomes. And to see more closely what's going on, we can describe the physical characteristic, the trait that's being observed here, and that would be the phenotype. So phenotype refers to physical characteristic or observable trait, and that obviously is depending on the genetic makeup or genotype that's driving that genetic trait and its expression, right? And so genotype describes the allele composition at a given gene. It's your genetic makeup. Your phenotype depends on genotype. So in this case, with our purple and white flowers, we have the phenotype of being either purple or white flowered, but those phenotypes are determined by the corresponding genotypes that we just saw playing out in our cross. So the phenotype ratio that was observed in that F2 generation was 3:1, but that was being driven by genotype ratio of 1:2:1, as we saw in that Punnett square, 1 out of 4 being individuals that have two alleles the same type, both the dominant purple allele. So we can define that state of having two alleles of the same type as being homozygous. So your two alleles are the same. That's homozygous. Your two alleles are different would be the heterozygous state. And then down here again, we have homozygous once again. Two alleles are the same, but now we can distinguish between the state of having being homozygous dominant, in which the two alleles are the same and they're both the dominant allele, or homozygous recessive, in which your two alleles are the same but they're both the recessive allele, right? So phenotype is driven by genotype. Our 3:1 phenotype ratio with this sort of cross is being driven by a 1:2:1 genotype ratio. Right, and again, the reason that Mendel was able to determine that this was what was going on is by observing that 3:1 ratio and figuring out mathematically what could possibly account for it. And that's what we saw here in this F2 generation. Random combinations of two different allele types coming from two parent plants combining in all possible combinations. That's the way we get this 3:1 ratio. All right, so let's say that you're out walking in Mendel's garden and you observe a white flowered plant. If you observe a white flowered pea plant, would you know what the genotype of that plant is? Well, yes, you would, because you know that the only way that you can end up with this white flowered phenotype is if you have this homozygous recessive genotype. That's the only genotype that results in the white flowered trait. So that's unambiguous. On the other hand, if you saw a purple flowered plant, would you know what the plant's genotype was? Well, no, you wouldn't, because this dominant phenotype can be associated with two different genotypes, either the homozygous dominant genotype or the heterozygous genotype. You only need one purple flowered allele, so one purple flowered allele, to show the dominant purple flowered phenotype. The other allele can be either the dominant or recessive allele, right? So if you saw a purple flowered plant, how would you determine the genotype? So if purple can be either homozygous dominant or heterozygous, how would you determine which was which? Well, you could carry out multiple crosses across the generation between purple flowered plants and try to work it out that way. But if you actually do one controlled experiment, given the fact that pea plants give hundreds of offspring in each cross, you could actually answer this question with one genetic cross. And that would be a cross in which you take your unknown individual, right? Your unknown organism that's showing the dominant phenotype, but you don't know what the genotype is. It could be homozygous dominant or heterozygous, and cross that unknown plant against a known plant that has the recessive genotype, which we know is associated with the state of being homozygous recessive, right? So we know what the genetic contribution from this recessive phenotype plant is going to be. Every single one of the gametes produced by that plant is going to have the recessive white flower color allele. And the phenotype that we observe in the next generation is really going to be determined by the gametes coming from this unknown plant's gametes, right? Whether they're passing on the purple allele or the white allele. And that's going to be dependent on what their genotype is. So we have two possible scenarios. Under scenario 1, our unknown parent plant is homozygous dominant and can produce only purple flower allele gametes, right? That's all they've got, that's all they can pass on. The other scenario, if that purple flowered plant was heterozygous, they would be able to produce two different types of gametes, purple allele gametes and white allele gametes. And so the offspring that we would get under scenario 1 or scenario 2 are going to be different from one another. So if this purple flowered plant was homozygous dominant, and all of the gametes they produce are going to have the purple flowered allele, then when these are combined through random fertilization with those white allele gametes coming from our white flowered parent, our recessive parent, then we are going to get all 100% heterozygous offspring. And we know that that is going to result in the phenotype, the physical characteristic of being purple. So if our unknown purple flowered plant was homozygous dominant for the purple flower allele, then we would expect to see 100% of the offspring produced in this cross showing the purple flower color trait. On the other hand, if that purple flowered plant was heterozygous and could produce two different allele types, when we combine those in all possible combinations with the alleles being transmitted via the gametes coming from our white flowered plant, then what we would see is 50% of the offspring are going to be heterozygous and therefore purple flowered, and 50% are going to be homozygous recessive, which we know is associated with that recessive phenotype of being white flowered. So by looking at the offspring that we get from carrying out a cross between our unknown plant and a plant showing the recessive phenotype, which we know is associated with the homozygous recessive genotype, looking at the offspring that are produced, we can determine whether the genotype was homozygous dominant or heterozygous and figure it out that way, right? Now that only works if you're generating enough progeny that you can be reasonably certain that you're seeing all possible genotypes represented. Obviously, if you're dealing with, for example, an experimental cross between animals that only have one or two offspring, that wouldn't work. But because pea plants provide enough offspring that you can be statistically reasonably certain of the representation of each genotype that you're seeing, we can use this one cross strategy. So that's referred to as a test cross. You're testing the genotype of this unknown plant by crossing it to a homozygous individual. So it's used to determine the genotype of an individual showing the dominant phenotype when that can be driven by multiple genotypes. Okay, so what we've been talking about here is a cross involving one gene with two contrasting alleles, right? So one gene, the gene determining flower color, and we've got two contrasting alleles being inherited here, the purple flower allele versus the white flower allele. This is referred to as a monohybrid cross. You're creating a hybrid generation for only one gene, right? And so Mendel's conclusion following his experiments with these monohybrid crosses was no blending inheritance. We did not see a blending of white and purple. We saw these two genetically inherited characteristics being maintained in their original state across the generations. You might see one trait recede into the background, but then it would reappear, right? So no blending inheritance. So Mendel answered his first question. The second question that he had turned his attention to was one that was a bit more complicated, and that's the question of whether parental traits are always inherited together. So, for example, if we're looking at two different traits now, seed color, yellow versus green, and seed shape, round versus wrinkled, right? So round and smooth versus wrinkled. And we started with two true breeding, once again, true breeding parental strains that have either yellow smooth seeds or green wrinkled seeds. Looking across the generations, if we followed the inheritance of these traits, would we always see smooth and yellow being inherited together? Would we always see green and wrinkled being inherited down the generation? So that was the question that Mendel wanted to address next. So here we're looking at the creation of a hybrid between two true breeding strains for not one but two genetic traits. And this is the most simple scenario where each gene has two possible alleles each. So this is referred to as a dihybrid cross. All right, so let's think about this. So if we cross a true breeding parental strain that's smooth yellow with a true breeding parental strain that is green wrinkled, and we symbolize those genes and their alleles with the letters S and Y, so S symbolizing either smooth or wrinkled, and Y symbolizing either yellow or green. Do we always see smooth and wrinkled being inherited together, green and wrinkled being inherited together? So when Mendel did this crossing these two true breeding parental strains, his F1 generation looked like this. It was 100% smooth and yellow, which provides us actually important insight into the way that these traits, yellow and green, smooth and wrinkled, interact with one another and the dominance relationship between them. Because the hybrids, which contain one allele of each type for each gene, right? So a smooth allele, a wrinkled allele, a yellow allele, and a green allele. And what we see is smooth and yellow. This tells us that smooth and yellow are dominant over wrinkled and green. So wrinkled and green are the recessive traits, smooth and yellow are the dominant traits. Now let's do the thought experiment. If these traits, smooth and yellow and green and wrinkled, were always inherited together, what types of gametes would this F1 generation plant be able to produce, and what offspring ratios would that result in the next F2 generation? So to say that again, let's say, so we know that this parental plant passed on in its gametes. Every single gamete looked like this. It had one allele, and I'm just drawing a line underneath these lowercase letters to make sure we're distinguishing them from the uppercase letters here. Each and every gamete produced by this yellow, smooth parent plant has an allele that specifies smooth and an allele that specifies yellow. So these are two different genes. Each gene is represented once in the gamete, and because the only allele for the smooth versus wrinkled gene this parent plant has is the smooth allele, that's what gets passed on. Likewise for the yellow, right? And then the converse for our green wrinkled parent plant. So if smooth and yellow were always inherited together, then we would assume that they would be transmitted together through this offspring, right? And so this offspring would likewise be able to produce two gamete types now. One gamete type would be smooth yellow. The other gamete type would have alleles for wrinkled and green. So if they were always inherited together, then if we intercrossed this F1 generation and thought about the types of offspring ratios that we would get by random fertilization between every possible gamete type, I forgot my underlines over here, then what would we expect to get? We would expect to get in our Punnett square exactly the same ratio that we observed in the monohybrid cross, right? We would expect to see a 3-to-1 ratio of yellow smooth and green wrinkled. That's what we would expect. So if these traits were always inherited together, then we would expect for our phenotypes a 3-to-1 ratio of smooth and yellow to green and wrinkled. And that's under the assumption that smooth and yellow always go together, green and wrinkled always go together. So that's the thought experiment. That's what we would expect. We'd have to do the experiment to see. But what would we expect if those parental traits are not inherited together? So they're not inherited together. Well, if they're not inherited together, then that means when this organism in the F1 generation produces gametes through meiosis, we're going to have independent assortment of the S gene from the Y gene. And this dominant S allele could segregate with the dominant Y allele, or it could segregate with the recessive Y allele. And so we're going to end up with gametes that look like this: dominant allele, dominant allele, dominant allele, recessive allele. And likewise, for the recessive allele from the S gene, it could segregate with the dominant Y allele, or it could segregate with the recessive Y allele. And so we're going to end up with those two gamete types. So if they don't always go together, dominant, dominant, recessive, recessive, they can assort independently, then we expect these F1 generation plants to be able to produce not 2 but 4 gamete types, right? And if we created a Punnett square in which we put the gamete types down the side, and remember that what we're doing is we're intercrossing these F1 plants with each other, so they're all going to be genetically identical, they're all going to produce the same types of gametes, then what we would end up with is a 4 by 4 Punnett square. And if we go ahead and draw out all of the genotypes that we would get from combining these gametes in all possible combinations, then we'd get 16 squares here. And I'm not going to go through and draw these all out because they're in the next slide, but just so that you see what we're doing here, this gamete combined with this gamete produces an offspring with this genotype, right? Homozygous dominant for the S gene, homozygous dominant for the Y gene. In this case, we have a recessive allele coming from this gamete for the Y gene, and so our genotype would be homozygous dominant for the S gene, but heterozygous for the Y gene, right? And so if we go across and we complete all of these rows, then what we would see is a Punnett square with 16 combinations of genotypes from our gametes, sperm and egg, coming from those F1 plants that each produced 4 gamete types, and we get this grid of 16 possible combinations here. So that's what we would expect if these traits, smooth and yellow, green and wrinkled, don't assort together across the generations. So a lot more complexity in the genotypes and phenotypes that would be determined here, right? Because anywhere that we have dominant alleles for each of these genes, we're going to see the dominant trait. And so we observe offspring in the F2 generation that are smooth and yellow. We observe offspring in the F2 generation that are green and wrinkled, but we also would observe offspring in this generation that have new combinations of traits that weren't originally present in the parents. So wrinkled with yellow, smooth with green. That wasn't here, right? We didn't see that in the parental generation or the F1 generation. That's a new combination of these characteristics. So that's driven by the greater diversity in the genotypes that are possible with independent assortment and segregation of these alleles. So how would you answer the question then? That's what you expect if they don't assort together, if they don't get inherited together, and this is what you expect if they do. So you do the experiment and you see what you get. And Mendel did the experiment, and what he got was this outcome, of course. A 9/16 to 3/16 to 3/16 to 1/16 ratio. or what's referred to as our 9 to 3 to 3 to 1. But that's 9/16 of the offspring. We would expect to have the dominant phenotype for both. So 9/16 would be dominant for both. So remember, our dominant trait was yellow for the color and smooth for the shape. And so 9/16 of our offspring are going to be yellow and smooth. 3/16 of our offspring we would expect to be dominant for one, but recessive for the other. So green and smooth, for example. 3/16 are going to be dominant and recessive, but in the other combination. So recessive for the first, dominant for the second. So yellow and wrinkled, for example. And then 1/16 we would expect to show the recessive phenotype for both. And that, of course, is our green wrinkled seed down here at the bottom. So the outcome of Mendel's experiment in his dihybrid cross was not the same 3-to-1 ratio that he observed in the monohybrid cross. It was this 9 to 3 to 3 to 1 ratio in terms of phenotype, and so that allowed him to answer the question, are parental traits always inherited together? And the answer was unequivocally no. And we know, based on our discussions of the molecular biology that's driving gamete formation, that this is because of the independent and random alignment of different chromosomes during meiosis and their independent assortment from one another. So we refer to this second important experimental outcome that Mendel described as Mendel's second law, the law of independent assortment. Alleles of different genes assort independently of one another. Now, there are circumstances in which Mendel's second law would be violated, that alleles would not assort independently of one another. And I'll let you think about that for just a minute. We're going to address that question in a later video. But we should recognize that what Mendel's second law is predicated on is the inheritance of distinct genes, distinct genetic sequences on DNA molecules. And as we've talked about, eukaryotic organisms have multiple DNA molecules, multiple chromosomes. And so we have to recognize that the outcome that he observed, and that very straightforward independent segregation he observed because all of the traits that he was investigating were on different chromosomes. And if traits are on the same chromosome, we would actually not, or possibly not, see independent assortment at all. We might see genes being inherited together because they're physically on the same bit of DNA. And we'll come back to that later and talk about that in the second genetics video, where we'll flesh that out a little bit more and think about so-called linked genes that are present on the same chromosome and how they're inherited. So Mendel's first law, the law of segregation, and Mendel's second law, the law of independent assortment, he didn't know what the molecular explanation for those laws was, but we do, right? We know what DNA is. We know what chromosomes are. We know what the molecular events of meiosis are. And so we can actually flesh out these laws of inheritance a little bit more and actually link them to molecular events. So let's think about Mendel's first law. What he called units of inheritance, that he described as being particulate, we know are physical DNA sequences on genes, so sequences of nucleotides that we call genes that are on these physical structures, DNA molecules. So they absolutely are particulate. They are physical structures that are inherited from one generation to the next. These alternate forms of these units of inheritance, as Mendel called them, these alternate forms of a gene, we refer to as alleles, which we know arise from random mutation. Right? So we get new alleles generated in the population by differences in DNA sequence that arise from random mutation in the genome. And every single living organism is constantly accumulating random mutations, most of which we never see a physical characteristic associated with it because they are not maintained in the population, but some of them are. So when we say that each individual has two copies of each of these units of inheritance, we now know that that's referring to our diploid genetic makeup. Two copies. We inherit one copy of each chromosome from mom, one copy from dad, and we know that those two versions of the same chromosome with the same sets of genes are called homologs. And we know that the process of meiosis segregates these homologs so that each gamete only ends up with one of each chromosome type, not two, right? So we go from diploid germ cells to haploid gamete formation via the process of meiotic cell division, as we talked about before. So this is really thinking primarily about meiosis 1, right? Mendel's law of segregation is really describing the events of meiosis 1, even though he never knew anything about DNA or meiosis. So we can go back and link the outcome of his experiments when he saw that purple flower colored trait and that white flower colored trait, that those were actually a gene specifying flower color, and we had alternate versions of that gene on the two homologs inherited from the two parents, right? So these would be alternate alleles at the flower color gene locus or position in the chromosome. All right, what about his second law? Well, Mendel's second law of inheritance is describing the behavior of homologous chromosomes and chromosome pairs with respect to other homologous chromosome pairs during meiosis and the way that they align during metaphase of meiosis 1. So remember that we said that these pairs of homologs or tetrads align in two possible orientations. So here we see a germ cell undergoing meiosis in which the homologs for the S gene have aligned in such a way that this chromosome carrying the dominant allele is going to be going up during this segregation. This chromosome containing the recessive allele is going to be going down. And just by random chance, we just so happen to see that the chromosome containing the Y allele is going to go the same way as the chromosome containing the dominant S allele, right? In this particular alignment possibility. But what we have to recognize is that other germ cells in that reproductive organ are going to be aligning with an opposite orientation. So I'm not going to draw the chromosomes, I'm just going to draw the letters. But in that opposite orientation, we would see the dominant homolog for the S gene aligning on the same side as the recessive homolog for the Y gene. And so our dominant Y homolog would align on the same side as our recessive S. And so under this scenario, dominant Y and recessive S are going to go together, dominant S, recessive Y are going to go together, whereas under this scenario, dominant S, dominant Y go together, recessive S, recessive Y go together. And so in some germ cells, we're going to have this alignment pattern. In other germ cells, we're going to have this alignment pattern. And if we look at the gamete population as a whole, they're going to be the outcome of both of these potential alignment configurations. And so this dominant S, dominant Y, and recessive S, recessive Y gamete came from that alignment configuration. This recessive S, dominant Y, dominant S, recessive Y came from this alignment configuration, right? So random alignment of our homologs at metaphase 1 leads to independent segregation of those chromosomes at anaphase. And we have to recognize that there are two possible ways for each homolog pair or tetrad to align, right? Two possible orientations for each one. And so that's where we get our second law. There's nothing that says dominant allele has to go the same way as recessive allele. They're equally likely to go opposite directions in that first meiotic division, which is going to give us different gamete types. So we get 4 potential types of haploid gametes, and that's what gave us our classic 9:3 to 3:1 phenotype ratio in our progeny.